She has published several research articles in reputed journals. The topic of today's session is cellular stress and cell death, how death signals flow inside the cell. So I request Miho Suzuki ma'am to please come up on the stage and deliver her thoughts. Please put your hands together and welcome ma'am on the stage.
Okay, I try. Oh. No signal? No signal. What? Okay. Okay, let's begin. Namaste. Thank you for giving me an opportunity to talk about my work. Actually, this is the second time to visit Biyani College, so I feel so nostalgic. Today's title is Cellular Stress and Cell Death. How does signal flow inside the cell? From biotechnology, so really different from the previous great presenter. But anyway, let's begin. In our body, cells constantly receive many signals or substances to respond for communication with each other and maintaining their homeostasis. To find molecules involved in responses to some inputs and rules governing the pathway, I have developed sensor molecules to detect them with non-invasive and high spatial in cell and time-resolvable approach. These are signal flow related to cell death due to some unfavorable signal inputs. Upon such signal coming, cells select a pathway to respond correctly to rescue themselves for survival or suicide. Surprisingly, they have so complicated judge points. This is an abbreviated signal flow. The arrow start side station represents cellular membrane, and the terminal station symbolizes nucleus. Like Tokyo subway map, it's so complicated, and they cross together at many points to exchange signal. After signal arrival at nucleus, cells might decide their responses, such as suicide, excretion of some factors, intake more energetic substances, or cell division to step forward. Then, how cells decide and answer to unfavorable or unavoidable attacks? Cell death might be very bad or final event for living organisms, but it could be correct partially. As for multicellular organisms, death in tiny portion of cell population may not damage so much to the individual. Multicellular organisms prepare several reactive patterns to such attack or stress. Here is well-known five well-known cell death types. Necrosis is distinctive because of its cause of death murder. The other one is a kind of suicide. Among cell death types, programmed cell death, a kind of cell suicide, is really attentive processes on the point of no bothering to other cellular activities consisting of the individual. Then, from those phenomena, I try to find some aspects of the nature of biological organisms. However, it is too puzzled to distinguish between different cell types, so classification will be still controversial. Apoptosis, most studied cell death, has a striking another function during biological development. For example, tadpoles lose their tail by apoptotic processes. Most transformation in biology involved in development may be caused by apoptosis. These inevitable activities in living organisms are very important, I think, but uncovering how to struggle to cellular stress would give a hint for some essence of living organisms as well. We have already known cell behaviors toward apoptosis consistent. The concept is based on no dispersing of disordered cell contents to our body before cleanup by phagocytosis. Though one such apoptotic aim could be fixed, there are so many complex pathways shown here according to stress inputs like this. In most study cases, one tries to know which routes or what molecules are included during respective apoptosis, or how cells determine their fates. In my case, a little bit different from their purposes. Attempts for revealing rules in trigger factors between upstream and downstream events. Surprisingly, even clone cells have their own properties. So I usually treat already established upstream and downstream events to accomplish my goal. As my strategy are living cell imaging, so I skip common biochemical methods such as electrophoresis, mass spectroscopy, etc. I only monitor fluorescent profiles emitted by sensor molecules introduced into alive cells. Then, what is fluorescence? And before that, what is light? Light represents not only visible light, but also invisible light, such as X-ray, radiation, UV, infrared, microwave, etc. Light is scientifically defined as energetic particle called a photon with wave performance. This wave and particle duality is very characteristic to light and should be comprehended to treat. To summarize the light, the shorter wavelength, the higher power, the vice versa. And visible light is located between UV and infrared, the middle energy. Here are photos stain cells with fluorescent dye. They give us information about the static conditions of cells. And these patterns represent genetic sequence analysis decoded using sequencer with also fluorescent dyes. Then, what is fluorescence? Fluorescence is light emitted from substances excited with light. So, excitation light is transferred to another light, fluorescence. By the way, firefly or sea firefly emit bioluminescence, not fluorescence, because their lights are produced by chemical energy, not light energy. Actually, I need two fluorescent substances because I should normalize any biological phenomena, such as enzymatic activities, pH exchanges, protein-protein interaction, into one fluorescence measurement. So I utilize fluorescence resonance energy transfer, called FRET, defined by Dr. Felster. When I use his theory, I get information about biological event by measuring changes in the ratio of two fluorescence intensity. Moreover, when FRET occurs, fluorescence lifetime, emitting time of one fluorescent molecule, will decay. We can detect this by measuring fluorescence lifetime directly. The reduction rate is almost equal to FRET changes. In fact, Professor Streyer, very famous biochemist, he wrote a lot of books or textbooks about biochemistry, put forward to use FRET to study various biological phenomena occurring in living cells. Fluorescent substances don't prefer materials. We have a lot of fluorescent stuffs. For example, fluorescent protein, fluorescent organic compounds, and quantum dot. It's a semiconductor nanocrystals and fluorescent polymer, etc. So I have picked up two different fluorescent materials for sensor molecules. Connecting mode of two fluorescent substances is essential for the good FRET sensor molecules. And this is a three-dimensional structure of the famous green fluorescent protein GFP. I take advantages of genetic manipulation to generate GFP mutant more suitable for FRET pair. Furthermore, I have inserted some sensing region into GFP sequence with the same methods. Fluorescent organic dyes, in most cases, are supplied here, shown here, as derivative forms to make bonds specifically to some functional groups. So it's nice. Semiconductor nanocrystal Q dots are usually covered with some organic polymers here for hydrophilic property and further chemical reactions. These are efficient FRET patterns between representative fluorescent protein as BFP, blue, GFP, green, YFP, yellow, and RFP, red, and organic fluorescent dyes. All blue line represent emission profiles of no FRET fluorescent protein. Here, fluorescent lifetime decay table of fluorescent proteins in FRET pairs of the previous slide. GFP has about 2.6 nanosecond fluorescent lifetime. The lifetime decay into less than 1 nanosecond upon FRET. Then I have prepared two protease sensor molecules using green and red fluorescent proteins. Proteases are caspase-9 and caspase-3, very important proteases functioning upon apoptosis. I can measure protease activity through FRET broken fractions. And these are FRET pattern changes upon digestion with caspase-3 in test tubes using various combinations of two fluorescent molecules. And these bar graphs stand for normalized enzymatic activities obtained by all measurement of sensor molecules with various combinations. It would be so quantitative. Here, these are calculated table for Phillips-style radius R0 of sensor molecules for caspase-3 detection, shown in the previous slides. This number with prefix A represents a very popular fluorescent dyes type commercially available. Almost combinations have large Phillips-style radius, meaning guarantee of efficient FRET in any combination. Then I introduced the sensor molecule into the cells using protein delivery reagent. The uptake mechanism might be natural endocytosis. Here is the live cell imaging to monitor caspase-3 activation with time and space resolving manner upon apoptotic inductions. Next, after two single index, caspase-9 and caspase-3 monitoring, I carried out double index monitoring in a single cell, yes, shown here, with cartoon. The time lapse of both sensor molecules imaging demonstrated caspase-3 activation occurred, followed caspase-9 activation, and activations were fully locally regulated. This experiment is not so elaborate, so corresponding cell experiments in large quantities were easily done. Here are individual cell response patterns. Absolute response time and intensity were totally different in individual cells. Surprisingly, these are all obtained from cloned cells. Anyway, I achieved statistical analysis for these data. Interestingly, this is a very beautiful average pattern. And I finally found relationship over individuality of cells between Caspase-9 integration, meaning activation intensities and continuations, and initiation time points of Caspase-3 activation. Through the same treatment of cells, I could use more powerful static tool called FACS and fluorescence lifetime analysis. What is FACS? FACS is fluorescence activated cell sorter. FACS analyzes cells spectroscopically at high speed, about 50,000 cells per minute. These are typical analyzed data of apoptotic cell. One dot represent one cell fluorescent properties. FACS functions not only high-speed optical analysis, but also versatile data analysis at a time. So, time course of fluorescence lifetime population changes upon apoptotic induction were examined by FACS. And I arranged fluorescence lifetime change by 3 experimental trials and analysis based on a histogram demonstrated in the previous slide. The order of Caspase-9 and Caspase-3 activation here was coincided with microscopic data. Dose-dependent manner of apoptotic induction drugs were tried as well. This is also average of 3 separate trials of dose dependency of apoptotic induction. Here, I got consistent data of relationship between Caspase-9 and Caspase-3. So, statistical analysis of high-content imaging and FACS analysis would render the partial rules of upstream and downstream of signal flow. This might be one of answer how to decide responsives to move or stay during various signal flow, not limited as stress responses. So I will expand monitoring indexes to access a general rule regulating such signal flow. Thank you for your attention.
Thank you very much, ma'am. Thank you very much, ma'am. It was very informational session about the general rule to govern apoptic pathway. 2 FRET-based chimeric sensor molecule to detect Caspase-9 and 3 activities that are the key enzymes for apoptic pathway were introduced into single cell for monitoring apoptic signal flow by initiation and saturation time. It was really very informative. Now it's time for the queries. If any of the student or the audience present over here want to ask, they may ask, ma'am.
Hello, ma'm. Ma'am, there is a question. What is the role of Caspase-9 and 3 and how they work?
Yes, that's an important question. Caspase-9 might be an initiator of apoptosis, and Caspase-3 might be an executor of its apoptosis. So if when Caspase-3 is activated, no return. Thank you.
Yeah, we have another one in the back side.
Good afternoon, ma'am.
Good afternoon.
Ma'am, my question.
Please use mic, please.
Difference between cell death and apoptosis, I guess. The difference between cell death and apoptosis.
Ma'am. Ma'am, what is the difference between cell death and apoptosis, and how we can identify cells experimentally?
Yes, that's also an important question. Anyway, it is still controversial to discriminate the cell-death type, but we definitely discriminate programmed cell-death and necrosis. Necrosis only avoidable death. But in other case, four case of cell death might be a programmed cell death. Really thoughtful death. But we still know, we don't still know which molecule or which pathway used for each cell death. But it might be a very significant for cell, but we don't know exactly now.
Do we have any other? So, let me have one question for all of you. It's, I think, very nice to hear this sensing, FRET-based sensing technology. Professor Suzuki, can you let us know how our students can use your technology when we don't have enough resource offsetting in our, so in low resource setting, how we can use your FRET sensor? Is it possible to use the FRET sensor in low resource setting?
Yes, sure, definitely.
Where we don't have much facilities, so how we can use?
It's simple because we need to detect only fluorescence change, so it's very simple. No elaborate equipment. So anyway, fluorescence spectroscopy or in many cases, fluorescence microscopy might be needed. Yeah, but it's not so expensive. I think it's not unusual. It's a really very common equipment you need to detect. Yeah.
So I think this is very good for all of you. If you want to use this FRET sensor for your cell death or cell apoptosis, you can use it, because also I think the Cambridge, some researcher in Cambridge has developed a paper-based microscope. If you haven't heard about we can also arrange the fluorescence microscope in low resource setting. So it's up to you. You should know the technology, so you can use this technology in your small setup of laboratories. So thank you again,
thank you,
Prof. Suzuki, for sharing your knowledge. Thank you very much.